Hello students, today we are going to talk about something called as dosage compensation. As we all know that this dosage compensations come under the subtopic of your sex determination only. About the sex determination, we have discussed this sex determination will be taken by three methods, like we have discussed. One of the method is you having sex chromosomes like X and Y. Another one will be your the ratio of your autosomes to sex chromosomes. Then the third will be your sex determination taking place by environmental factors, like taking place into tortoise and like you can say crocodile. In these things you will having the sex determination by the environmental impact. So why this dosage compensation is needed? If we'll see this dosage compensation is needed because you know that the female is having two X chromosomes and male is going to have only one X chromosome and another Y chromosome. Okay, so what happens over here, as we have discussed, according to your human genome project, your Y chromosomes carrying only 73 functional genes, whereas your X chromosome is going to carry 2800 to 1400 genes, something around. Okay, they are going to carry about something called as your 900 to 1400 genes. Now if we'll compare the amount of genetic pool or amount of genes which is carried by a female and a male will be the different. Okay, over here if we'll consider the female having two X chromosomes is going to have around 2800 genes expressing at the same time, whereas in the female it is going to, whereas into the male it is going to having only you can say about 1473 or something. So this dosage differentiation is needed to be compensated in both the male and females. Basically this dosage compensation take place into the female most of the time. Okay, what happens into the dosage compensation, as you can see over here, in order to compensate the doses of the genes present on the X chromosome, one of the X chromosomes into the female have to be inactivated. So that is why it is got inactivated, and this inactivation is taking place into the early development. Okay, early development stages like that will be taking place into the either into the morula stage or other into the blastocyst stage. In either of these cases they are going to be inactivated. Inactivation means they are going to have the metabolic activity, but they are not going to take part in any kind of gene expression. Only the genes are not going to be expressed. Okay, where in the cell division they are going to be passed on from the one say about if there is a mitosis or the meiosis, not into the meiosis but basically into the mitosis. If they have to divide, if the cells are dividing, obviously they are going to pass on from the one cell to the another cell. Okay, so the dosage compensation is nothing but it is the order, or it is the order in which your doses are getting compensated of the X chromosome, so that your one of the X chromosome basically into the female is getting deactivated or inactive in the scenario. So why this is needed and what is happening over here is, see, this dosage compensation basically has been covered by or has been found out by the Murray Barr and Edward Barton. Observe that the darkly stained body in the interphase nerve cells. Okay, why this is going to be into the interphase nerve cells? Because you know that such chromosomes are going to be expressed into your mitotic phases only when you'll have the cell division. But how does this chromatin bodies has been come up into the interphase? Because they have got inactivated. Okay, they are not going to take part into the other things. Okay, that is why this Murray Barr and Edward Barton has observed a darkly stained body in the interphase nerve cells of the female cat that was absent into the sphincter of the male. So they are present into the females, but they were absent into the males. This given them a sign that there is something which is present into the female but absent into the male, so that in the humans they have find it out the same thing demonstrated into the female derives them from the buccal mucosa cavity, chick cells, and the fibroblast, and that is not present into the males as well. Okay, and these structure they have called it as sex chromatin bodies. These sex chromatin bodies are also called as Barr bodies. Okay, so 6 chromatin bodies are also called as the Barr bodies and is an inactivated X chromosome. Shamshu On is one of the scientists who have found out that and who have first suggested the Barr body arises from one of the two X chromosomes present into the female. Okay, so if one of the X chromosome is inactive, in the cells of the female, the doses of the genetic information that can be expressed in the male and female will be equivalent. As we have discussed, sir, आपके पास X chromosome में you have something called as 1400 genes, whereas Y chromosome is having only 73 genes. So to compensate these genes come out, one of the X chromosome itself get inactivated and another will be expressing itself. So the amount of genetic content in your X chromosome and Y chromosome will be the same, around something called as equivalent, because male is going to have something called as 1473 genes and female is going to have 1400 genes. But the 73 genes of the male we have already discussed, they are hemizygous in nature. Okay, so obviously they are not going to take part in anywhere. So these bar bodies, regardless of how many X chromosomes you have, okay, in the somatic cell, like we have discussed, there are diseases like Klinefelter hair, you will have other diseases where you have Turner syndrome, where you have either you will have only one X chromosome, or you are going to have 2X chromosome, or 3X chromosome, or 4X chromosome. Regardless of the number of X chromosome, what is going to happen over here, you are going to have only one X chromosome expressed, and other X chromosomes are going to be inactivated. Okay, for an example, over here you can see that first one, no bar body is seen into the somatic cell of the Turner syndrome. Why? Because they are going to have only one X chromosome. So obviously, since they have only one X chromosome, so they are not going to have any formation of the bar body. Whereas the second one, if you'll say the Klinefelter wala condition or the normal condition as well, where you will have the two X chromosomes, that is XXY or double X in the female, you are going to have only one bar body. 2 bar body you are going to find it out where you will have triple X. Okay, and agar aapke paas 4, if you have the 4X chromosomes into the female, they are going to have 3 bar bodies. So the bar bodies, the rule follows over here is it N1 rule is going to be followed up over here. Okay, so this is something called as, you can see over here 46 or 45 where you will have only X chromosome. So no bar body ka formation over here is going to take place. Now in the second condition, if you'll say you will have only 2X chromosomes, whether it will be the normal condition, that is with 46 or 47 double XY, you are going to have the 1 chromosome. If you will have the 3 or 4X chromosomes, you are going to have the 3 bar bodies, and if you are going to have 3 bar bodies, you are going to have, you are going to have 3X chromosomes, then you are going to have the 2 bar bodies over here. This is how it is falling out. Now the big question come over here, sir, which of these X chromosomes are going to be inactivated? Because one X chromosomes come from the male partner and the second one is coming from the female partner. So whether it is maternal or paternal, which one is going to be inactivated over here? So in the mammalian females, one X chromosome of the maternal origin and one is of your paternal. So this confusion has been divided and has been cleared by your Mary Loan and Lennon Russell independently propose a hypothesis that irrespective of whether they are coming from the male or the female partner, any of the X chromosome get inactivated. See, that is what it has been written, that the inactivation of the X chromosomes occurs randomly in the somatic cell, irrespective of whether they are coming from the parental origin or from the maternal origin. It is not needed. It can be inactivated from any of these. Okay. So the inactivation of X chromosomes occurs randomly in the somatic cell at a point early of the embryonic stage, most likely sometime during the blastocyst stage also. So once inactivation has occurred, all the decent cells are going to have the same X chromosome inactivated in their initial or the another progenitors. Hopefully this topic has been understood by all of you. We'll cover the